little champions, I welcome you all to Decode NCERT series, where today we are going to decode a 7-marker chapter from NCERT Biology, that is Biotechnology Principles and Processes. Now this is a chapter that is new, engaging, interesting, and for some not so fun. I definitely understand it, but let me tell you, this is for the first time that is studying about biotechnology. You might have not studied until your 12th grade, so that brings out the excitement. Okay, it's a very interesting chapter, and now if you study this chapter really well, the next chapter becomes all the more easy for you, that is biotechnology and its application. Now let me tell you one more interesting fact, that is, in my 11th and 12th I had it as my sub-subject, as the second subject. Okay, so I was bio with biotechnology, so for me it's all the more very interesting, and I hope that I could help you with this particular chapter. The thing that I need you to do is take out your NCERT. If you have sticky notes with you, take the sticky notes. Okay, whenever I tell you that this type of questions have come, please put the questions there. Okay, this type of questions have come. Now you can frame your own type of questions as well if you need, but at least during the final session when you are there to revise, guys, you can just go through the questions and their answers which is given in the paragraph itself. Yeah, so it becomes easy for you. Do not underline, just like how I have mentioned over here. It's just for making you understand that this paragraph is important, that's all. So do not keep on underlining when I am talking to you. As of now, your heart, soul, mind, everything should be here with me. Okay, listen to me carefully, that's all. Fine? Chalo. Let's get started. So this portions and all the introduction part, you just leave. Where is it going? Okay. So from here, principles of biotechnology, my dear children, go give a quick read. Okay. Now if you go down, you see something like this, that is origin of replication. I have made this paragraph underlined a bit. Okay, what is origin of replication? In a chromosome, there is a specific DNA sequence, what do you call it? Origin of replication, which is responsible for initiating replication. This is where the DNA polymerase comes and attaches, and then the whole process of DNA replication takes place. So without origin of replication, I cannot imagine anything to start its replication process. Okay, so we need an origin of replication, otherwise things are not going to work. Now children, over here they have told about plasmid. Now there's a question that have come, define plasmid. In your PU2 board examination, the question has come, define plasmid. So if you do not know what is plasmid, then what will you do? Basically children, in bacteria and all, what happens, they have their chromosomal DNA that is responsible for coding proteins, and these proteins make what they are. But apart from that, there are certain extra chromosomal DNA also which are circular in shape, and they're just lying there. Okay, so these extracellular as in something that is extra in a cell, right? This extracellular autonomously replicating, as in they also replicate by their own, because they have what? Origin of replication. That is why they are replicating. So it's just there. So whatever changes we want to make, we can make in this particular plasmid. You get it? And we can make copies also, since anyway this plasmid is going to create a copy for itself. So why not put our DNA or gene of interest and make copies of it? So that is why plasmid is really important. So that is why I said you have to go through this definition of plasmid, my dear children. So now over here there is one more thing that they are given, the construction of the first recombinant DNA. What do you mean by recombinant? For example, there is already pre-existing some type of thing. Now you are putting your thing over there. Don't you think you are recombining something which was already combined? You are making a new combination over there. This is known as recombinant, and when it's talking about DNA, you call it recombinant DNA, new combination of what? DNA. So construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid. So what do? A gene was there which was giving antibiotic resistance. For example, I am having the gene for antibiotic resistance. That means even if I have that antibiotic, there won't be anything that will be happening in my body. I will be resistant to that particular antibiotic. Whatever bacteria is there in my body, it will still thrive anyways, because my body is resistant to that particular antibiotic. Okay, that is what you mean by antibiotic. Now we are cutting that antibiotic gene and you are putting it somewhere else. So let's see. So with a native plasmid of Salmonella typhi, Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistant gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. So they cut it, okay, this antibiotic resistant gene, and then they did what, children? They put this, when this DNA is transferred into Escherichia coli, that is E. coli, a bacterium closely related to Salmonella, It could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to make multiple copies of antibiotic resistant gene in E. coli was known as cloning. My dear children, what happened? So anyways, the antibiotic resistant gene, I cannot put it in Salmonella itself because Salmonella will get that gene. Salmonella typhi is the one that causes typhoid. So if I put the antibiotic resistant gene into that, don't you think it will be resistant? So whatever antibiotic we treat it with, it won't die. So what did we do? We put the antibiotic resistant gene just to check whether it's working. We put it inside E. coli. Okay, inside E. coli we put it, and then we put antibiotic and saw that it was able to live. That means this antibiotic resistant gene was really useful, right? You get it? So this is a new combination that was being formed, and also with the help of E. coli's replication machinery, it was able to create multiple copies of this particular antibiotic resistant gene as well. You get it? And this process is known as cloning. Similarly, for example, in my body there is a protein that has to be made, okay, but I cannot rely on my body. So what do? I put it in some other organism, for example E. coli. I put it in their plasmid, okay, the gene of interest, and ask them to replicate. Whatever they replicate, they keep on replicating, create multiple copies. I get more and more protein. Now I can take the protein whenever I need it. You get it? This is why it is very much useful. Now my dear children, when I said you have to cut the DNA, how do you cut it? With the help of the molecular scissors. Why do we call it molecular scissors? Molecular scissors as in they are going to cut the molecules of something. For example, over here they are going to cut the DNA. And what are the molecular scissors of a body, my dear children? It is restriction enzymes. What do you call them? Restriction enzymes. So you are going to cut the DNA with the help of restriction enzymes. Now these DNA, for example, the DNA that I am looking forward to, the DNA which is already there, it has to stick to each other. How do you stick it? With the help of the enzyme DNA ligase. So DNA ligase which act on the cut DNA molecules and join their ends. Now one more very important thing, these plasmid DNA acts as a vector to transfer the piece of DNA attached to it. So sometimes we say, what do you mean by vectors? Vectors are something that will be helping me in transferring something. Okay. So for example, I have the DNA with me, but I have to transfer it to an organism. How do I do it? Probably I insert this DNA into some other organism, and this organism will go infect another organism and give it anyways. So this organism which comes in between, who is responsible for passing the DNA, what do I call it? I call them as vector. Okay, what do I call them? Vector. So your basics of this particular chapter has been put over here in the in 164 page number, my dear children. Just go through it. It's very important. The basics is given. Without understanding the basics, you might not be able to understand anything else as well. Okay, now they have spoke about restriction enzyme very clearly. What do you mean by restriction enzyme? They are the molecular scissors, the one that cut the DNA. Now over here, in 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. So what was happening, my dear children? Escherichia coli is there, a bacteria, in which someone was trying to grow. That is who? Bacteriophage. Bacteriophage is a virus, was trying to grow. But then E. coli had two enzymes. E. coli produced two enzymes which were not allowing the growth of bacteriophage in their body. One of these added methyl groups to DNA while the other cut the DNA. So one of them, they added the methyl group to the DNA. Okay, so E. coli had its own DNA. Okay, the bacteriophage which came in, it also brought in its genetic material, probably converted into DNA also. Anyways, so this guy was there. If the bacteriophage put their genetic material into mine, that is I am Escherichia coli, into mine, then what will happen? It will be able to produce the proteins with the help of my enzymes and everything. I don't want to do that. So what do? I release two enzymes. One enzyme goes and cuts their DNA. One enzyme goes and cuts their genetic material. But I have to make my genetic material also safe. Otherwise, these enzymes which I am producing could cut mine also. So what do? I add methyl group and say that, hey, the one which has methyl group, you don't have to touch that. The one does not have, go cut it. So that is the thing. Okay, so basically in 1963 the two enzymes were recognized, and the later one, the second one, which was responsible for cutting the DNA, it was known as restriction endonuclease. What do you call it? Restriction endonuclease. So in Escherichia coli in the year 1963 for the first time you found restriction endonuclease. Okay, the restriction enzymes are divided into two parts, restriction endonuclease and restriction exonuclease. You will study about it. Now the first restriction endonuclease, HIND2. What is the first restriction endonuclease? Direct question. HIND2, my dear children. HIND2. It was found that HIND2 always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. So it had a 6 base pairs fixed. Okay, it had, oh my god, my tongue. It had it fixed, okay, not fixed, fixed, okay. Now, whenever the Hind2 sees these 6 base pairs, then only it will make a cut. So, what do I call this? I call this site as recognition site for the enzyme Hind2. Now, the question has come, Hind2 cuts DNA molecule by recognizing a specific recognition sequence of how many base pairs? 6 base pairs. How many base pairs? 6 base pairs. Please remember the question has come. Okay. Now, how do you name it? For example, Hind2 or whatever is there. EcoR1 is there. So, the first letter of the name comes from the genus, okay, that is EcoR1. E stands for Escherichia, okay, and the second 2 letters, Eco, CO, Co, come from coli, Escherichia coli, okay, comes from the species of the prokaryotic cell from which they were isolated. Now, EcoR1 comes from Escherichia coli, RY13. So, R comes from the strain, and 1, 2, 3, whatever is this is along with how they were discovered, okay, in which order they were discovered. You are giving the name EcoR1, EcoR2, EcoR3, things like that. So, E for genus, okay, and CO for the species name, the 2 letters of the species name you pick it up, and R for the strain's name, and 1, 2, 3, whatever you, it's not from 1 from 13, okay, please don't take that. So, Escherichia coli R by13 was the strain from which we got EcoR1 enzyme. That is again a very famous enzyme. Now, my dear children, enzymes which remove nucleotides from the ends of the DNA are, we don't know, ma'am. So, listen, restriction enzyme belong to a larger class of enzymes called nucleases. Okay, so restriction enzyme belongs to what? Nucleases. Nucleases as in the one that cuts nucleic acids, as in nucleic acid is what? RNA, DNA, everything acts as a nucleic acid, my dear children. Okay, so restriction enzymes comes under what? Nucleases, and there are 2 types of nucleases, exonucleases and endonucleases. Exonucleases remove nucleotides from the end of the DNA. So, enzymes that remove nucleotides from the end of the DNA are exonucleases, restriction exonucleases. And now, endonucleases make cuts at specific position within the DNA itself, at the specific position they make cut, and that is known as endonucleases. Now, each restriction endonucleases functions by inspecting the length of a DNA sequence. Okay, how do they work? Please remember, these are very important steps. They can ask you how do they work, so you have to tell. Okay, they do by restriction endonucleases, the one that cuts from the middle, okay, middle of the specific position within the DNA itself. So, over there, inspecting the length of the DNA sequence, they inspect the whole length of DNA sequence. Once it finds the specific recognition sequence, it will bind to the DNA and cut each of the 2 strands of the double helix at specific points in their sugar phosphate backbone. Where do they make the cut? Sugar phosphate backbone. DNA has sugar phosphate backbones, it will cut over there. So, it inspects the length. Once it finds the recognition site, it will make cut at specific points in their sugar phosphate backbone. Now, each restriction endonucleases recognizes a specific palindromic nucleotide sequence. What do you mean by palindromic? The one that is read similar in both the ways. For example, you have GAATTC. From this end also, if you read it, it will be GAATTC, my dear children. These are palindromic sequences, and always remember, restriction enzymes makes cut at palindromic sequences only. The recognition sites we are talking about, Hind2 has 6 and EcoR1 again has 6 only, okay. Where are they making cut? In their palindromic sequences, but specific location within the palindromic sequences. For example, the recognition sequence for EcoR1 is GAATTC. Please remember it, GAATTC. Okay, this is 5' end and this is 3' end. Similarly, this will be 5' end and this will be 3' end. Okay, now the thing is, okay, listen to me carefully. The thing is, here the cut is made between G and A. Similarly, here also the cut is made between G and A. Suppose there is one more DNA strand, okay, here also they have the same sequence. I make a cut between G and A. Here also I make a cut between G and A, my dear children. Okay, cool. I think it is mismatched. The arrow should be here. Okay, anyways, so they made the cut. Now it will look like this. They cut this, so it got removed. They cut this, so this portion basically it got removed. So what do you get? Something like this, an L-shaped thing like this. They made a cut over here, they made a cut over here. This L got removed. This L got removed. Similarly, here also if you see, there will be one L that is going to be getting removed. So this thing is removed. Now what do you have, children? This is free. This is also free. What are you generating over here? This thing that you see over here are known as sticky ends. What are those? Sticky ends, my dear children. They have the property to make hydrogen bonds with the next nucleotide. If they are same, if they find complementary nucleotides, they will be again making what? Hydrogen bonds between them. So these are complementary, right? For example, you see T is over here, A is over here, T binds with A, again T binds with A binds with T, A binds with T. So don't you think they can join together? And exactly that thing happens. They are going to join. So please remember, restriction enzymes which are there, which are going to be there, they cut in the recognition site. Okay. Now where the recognition site? In the sugar phosphate backbone, my dear children, and within the specific sequence they are going to make a cut. Okay, same cut, and they are going to generate sticky ends, and these sticky ends are going to join to each other as they have the property of forming what? Hydrogen bonds. Okay, and I hit my hand. Okay, anyways, sometimes when I get too energetic, something like this often happens, so it's okay. Okay, chota ho jao. Given below is a portion of the DNA strand giving the base sequence on the opposite strand. What is so special shown in it? 5-GAA-TTC, 3-, and then 5-GAA-TTC. What is so special in them? It is palindromic sequence, read same from both the end. 5-, this, 5-, this. So it's the same. It's known as palindromic sequence, my dear children. Now what is EcoR1? EcoR1 is a restriction endonuclease. It's a restriction endonuclease, not exonuclease. What is the sequence? You have to talk about that. GAA-TTC. 5-GAA-TTC, 3-. That's how you write it. Okay, it's an endonuclease which cuts the DNA. Now, how is the action of exonuclease different from that of the endonuclease? Exonuclease cuts at the end of the DNA, endonuclease cuts within the specific sites within the DNA, Now, how are sticky ends formed on a DNA strand? Why are they so called? They are known as sticky ends because they can make hydrogen bonds with the complementary base pairs if needed. Okay, so that is how they are formed and that's how they work. So, yes, that is important. Restriction enzymes cut the strand of DNA a little away from the center of the palindromic site. So, the palindromic site, that is GAA-TTC. So, restriction enzyme cut the strand of DNA a little away from the center of the palindrome. So, they are going to make a cut over here, that is between G and A, and they are going to make a cut over. Oh, this was not the one where I could write. Okay, we are going to make a cut over here, but between the same two bases on the opposite strands. So, here also between G and A, here also between G and A, my dear children. Cool. Now, when they cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends. So, only if they, when they are cut with the same restriction enzyme, then only you will have a common sticky end. Otherwise, you won't be finding. These can be joined together end to end using DNA ligases. Remember the name of the enzyme, DNA ligases. Cool. So, that's all. Please remember this diagram that you see over here is very important. Okay, so just make it if in a rough way if you want. In that way you will understand it a lot more better. And this is the thing that I was saying. For example, this is the foreign DNA. Okay, this is the vector plasmid that I am having. I made a cut over here and I made a cut over here. What I am going to do? I am going to put it over here. This is now a recombinant plasmid, my dear children. What do I call this? This is a recombinant plasmid. I use ligases to join them. Yeah, this recombinant plasmid, when I am putting it inside a bacteria, I will call this as transformed bacteria. This has underwent transformation because it got the recombinant one that I put, the new one that I put. So, it was what? Transformed. Or basically, whenever I put a plasmid apart from what they had, this is known as transformed. And a transformed one could be containing a recombinant one, or it might be not containing. Okay. For example, there is a plasmid like this, there is one more plasmid which did not undergo recombination. Both of them, if they are going to get in, you call it transformation only. The one that has the recombinant one, you call that cell as recombinant plasmid, say recombinant cell. Now, no. Okay. Okay, yeah. Now it's DNA fragmentation time. So listen to me very carefully. Okay, they have just made this chapter too complicated by writing everything here and there. So do not be terrified of it. I am going to make things really easy. If I move it entirely upar, I don't know. Chalo ek kaam karte. I'll move it entirely upar because I don't want to confuse you guys. Then you might think, what is ma'am teaching? Even ma'am don't know how to teach. Okay, wait. Okay. So this is the process of recombinant DNA technology. Okay, then we will come back to that particular page. This is page number 171, my dear children. So the first thing that you need to do is you have to isolate the DNA. Okay, Obviously, if you want to put some DNA in somewhere else, obviously you need to isolate it. So, isolation is not very easy because you know DNA is within the cell. So, cell wall might be there in some organism, you have to break down the cell wall. In some organism it's not there, then it's all cool. So, listen to me carefully. The DNA is enclosed within the membranes. We have to break the cells open to release the DNA along with other macromolecules such as RNA, protein, everything will be there. But we don't want anything, we just want the DNA. So, how do you isolate just the DNA? First thing, if it's a bacterial cell, my dear children, you have to break the cell wall made up of peptidoglycan. So, you use the enzyme lysozyme. These enzymes names are really important. Okay. So, use enzyme lysozyme to break down bacterial cell wall. Now, for plant cell you use cellulase, for fungal cell you use chitinase because it is made up, the cell wall is made up of chitin. Here the cell wall is made up of cellulose. So, that is why you are using these enzymes. So, now you broke the cell wall. Now, what happens, the RNA which is there inside, which is not needed by us, it also has to be removed. So, what do? We treat it with ribonuclease. Ribonuclease will cut down the RNA and all, we do not need it. Now, proteins can be removed by proteases. Okay, all these enzymes are needed. Now, ultimately you will get your DNA, and this DNA again there might be small chutru things that you have to separate it from. So, what do? You put chilled ethanol. When you put chilled ethanol, please listen to me carefully, all ears, okay. Chilled ethanol is very much important. When you put chilled ethanol, what will happen? The DNA which is there, it will get separated. Okay, it will get separated, and finally you can do spooling, you can take that thing outside, the DNA outside. So, only with the help of chilled ethanol, the DNA will be totally, settling down. Please remember that. Okay. So, this is step. These enzymes are really important. They ask you, I am telling it 10,000 times. So, please make sure you know about all of them. Now, you got the DNA. Okay. Choose the correct option for sequential steps of recombinant DNA technologies. The first step is isolation of DNA. Second step, cutting of DNA at specific location. How do we cut? With the help of restriction enzymes. So, how is your NCERT design? No, they have spoke about everything, the tools first, and then they spoke about the steps. So, they are not entirely long, but they explained way too much over there, so it might confuse you. So, what do you need? You need restriction enzymes, my dear children, to cut the DNA at specific location. You isolated it, you cut it now. So, restriction enzyme digestions are performed by incubating purified DNA molecules with the restriction enzyme. DNA is a negatively charged molecule, hence it moves to, okay. Okay. The joining of DNA involves several processes. After having cut the source DNA as well as vector DNA with a specific restriction enzyme, the cut out gene of interest from the source DNA and the cut vector with the spacer mixed and ligase is added. This results in the preparation of recombinant DNA. So, that much detail we have not to go, we do not have to go. Listen to me carefully. We cut the DNA, okay, the gene of interest we have cut, and also we have cut the vector from in where this particular thing has to be inserted with the same enzyme. That is done in cutting of DNA at specific location. So, isolation first done, then you have to cut it. So, you cut the DNA at specific location. Now, 1 or 2 might not be enough. So, we have to increase its number. We have to amplify the amount of DNA which is there. How do we amplify it? We amplify it with the help of PCR. What is it? Polymerase chain reaction, my dear children. Okay, what happens in PCR? Suppose this is a gene of interest that we are having. Okay, this is the gene of interest that we are having. The first step that it undergoes is denaturation, my dear children, but at high temperature, almost 90 to 95 degrees Celsius. At that particular point of time, what happens? Both the strands that are there, they will get separated. So, you see, separation has happened. The next step is annealing. The next step is annealing, where what is happening? One primer will come and attach over here, and one primer will come and attach over here. At both the ends, one primer will come and attach. Please remember, on the 3' side only this primer will come and attach. Similarly over here, why do you need a primer? If you have studied molecular basis of inheritance properly, you know for DNA polymerase to work, there should be initially some strand there, then only it can elongate. So, basically for DNA polymerase to come and act, whenever you want to make a copy, obviously you need DNA polymerase, right? So, before that you make a strand over here, that is known as primer. So, these primers are added and this is known as annealing, my dear children. Now, the next step, here the temperature can go a bit down, it is like around 50 degree, 52 something degrees Celsius, and then now you do extension. Now you do what? Extension. Extension again you have to take it up to 72 something degrees Celsius. At that point of time the DNA polymerase will increase, elongate on both the sides, and now you get what? From one strand you have got 2 strands now. In similar way the DNA is amplified again and again. So, these steps which are there, denaturation, annealing and extension, the sequence of this step is asked, the process has been asked clearly. Okay, you might have to write it in points describing what is denaturation, what is annealing, what is extension, or probably you might be asked the the sequence. Anyways, both of the answers you have to get it correct. Now let me tell you one very important thing, okay, one very important thing, that is you need DNA polymerase obviously for elongating, and also you need what? Deoxynucleotides. Certain nucleotides also you need, no, then only nucleotides are the ones that makes the DNA. Okay, so you need deoxynucleotides, the one that makes the DNA. So many small will come together, then that's how the DNA polymerase will attach each of them and form the new strand. Now DNA polymerase over here has given a special name that is known as stack polymerase. Okay, the DNA polymerase used in the process of PCR is given a special name that is known as stack polymerase, my dear children. T stands for thermus, AQ stands for aquaticus. So there's a bacteria thermus aquaticus which is there, which can thrive in very high temperature. So that from that particular thing the polymerase enzyme is isolated, which is why it is known as stack polymerase, which is why it is able to sustain high temperatures like this. Okay, please remember that. So choose the correct sequence of polymerase chain reaction steps from the following: annealing, denaturation, extension. These type of questions come, so you should be knowing denaturation is the first one, then comes annealing, and then comes extension. Okay, now going ahead, yeah, so over here sets of primers and synthesized, okay, 1 billion copies can be made, my dear children. DNA polymerases which remain active during the high temperature induce denaturation of double stranded DNA. Okay, listen to me, it is isolated from bacterium thermus aquaticus as mentioned. Okay, so now I isolated the DNA, I cut the DNA, I amplified, I made multiple copies of it. Now finally what do I need to do? I need to insert it into insert the recombinant DNA into the host organism, So there are several methods of introducing the ligated DNA into the recipient cell. Recipient cell, after making them competent to receive, take up DNA present in the surrounding. Okay, so I will be telling you how do you exactly put the DNA inside the host cell. Okay, do not worry about that, just know the steps. So we have finally put it in the host cell also. Imagine we have made the recombination happen, and then we have put it inside the host organism. Host organism is the one where it will produce its protein or whatever product it has to make, it will make there. Okay, so we inserted the recombinant DNA into the host cell. Now what will happen? Obviously it will give the product. It will ultimately give you what? The gene product. That is the next step it will give you. So the gene product have to be again you have to read the whole thing over here, okay, but let me tell you one very important thing, that the gene product, when it is isolated, there are so many other things also which comes along with it. So you have to make it really purified, okay, and this process where you are basically going to separate the product from everything else, you call this as downstream processing. What do you call this as? You call this as downstream processing. The processes include separation and purification, which are collectively referred to as downstream processing. What, children? Separation and purification. Separated and then purified. The product has to be formulated with certain preservatives as well. Whatever you get, you put it in certain preservatives. Such formulation has to undergo thorough clinical trials, and in case of drugs, strict quality control testing for each product is also required. So anyways, what do you mean by downstream processing? Separation and then purification. Separation and then purification. If needed, you add what that is known as downstream processing, and then you put what? You put preservatives as well, you do quality checking and things like that. So that is how things work: isolation, cutting, and then amplification, and then putting inside the host organism, and then basically extracting the gene product, and then basically cleaning it up. That's the thing. Okay, and one more thing I will tell you. Over here, the gene product, where is it made, my dear children? In a small scale things can be done, but in large scale we obviously need bioreactors. Okay, bioreactors are the ones which can carry volumes of around 100 to 1000 liters of cultures can be processed. So there is a question that has been asked about draw the neatly labeled diagram of a simple stirred tank bioreactor. They didn't mention simple stirred tank, but I am telling you, it's just this thing that you have to draw. Okay, you have to make sure that, okay, listen to me, the funda is that the host cells which are there, for example E. coli which is there, the E. coli is put over here, nutrients are given, oxygen is given, everything is given, such that this can increase their number in. Okay, and when they are increasing the number, they are also replicating the DNA which they are having, and it is having the gene of interest. So obviously they are increasing the product that we are looking forward for. You get it? So that is the use of bioreactors, and for that you need to provide them with proper pH, you need to give them a sterilized environment, you need to make sure that aeration is proper, that is all the parts of the bioreactor is getting air equally, okay, things like that. Okay, So that is the thing. A stir tank bioreactor is usually cylindrical or with a curved base to facilitate the mixing of a reactor contents. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Bioreactor has an agitator system as in the one that will help it move, an oxygen delivery system, a foam control system, a temperature control system, a pH control system, and a sampling port so that small volumes of culture can be withdrawn periodically. So while drawing, you should be making sure that you are labeling all this thing. You are labeling all this thing. Now this is parsed stir tank bioreactor that is not asked to draw, my dear children. The difference is that basically over here you insert the air with the help of bubbles. The help of bubbles, it becomes all the more good, better than this, but the picture of this particular thing is being asked because this is the normal one that we are having. Cool. So those are the steps, and now we'll go above again. Okay, where were we? So we were here where we spoke about restriction enzyme, and now it's time for the next thing. Now, separation and isolation of DNA fragments, my dear children. Once you have cut, it's not like, okay, once you have cut, again you have to make sure that the one that is cut, there are different fragments of DNA which is being cut, no? We have to make sure about the smallest fragment which is there, which is our gene of interest, that has to be taken up, and the gene that we are looking forward for. So how do you separate it? With the help of something that is known as gel electrophoresis. So since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards anode under the electrical field through a medium matrix. So there will be cathode, there will be anode. Anode is, my dear children, positive, and cathode is, my dear children, negative. Okay, so what do? You put the DNA over here. DNA are also negatively charged, cathode is also negatively charged. They will repel and they will move towards the opposite end, that is the anode. Please remember, DNA is negatively charged, DNA, N is there. You remember like that, N for negative charge. Okay. And anode is positively charged, remember like that also. Anions are negative, cations are positive, but here ulta is happening. Anode which is there, it is what? Positive, not negative. Please remember. So what will happen? The smaller fragments which are there, they will move forward. Yay, we are small, we will move forward, and then they will reach over here. Okay, they cannot easily separated. Now, so that is given over here. But one more thing, the DNA fragments separate according to their size through sieving effect provided by the agarose gel. What is the gel being used over here? Agarose gel, my dear children, and it is taken from the seaweed. It is taken from seaweed. The agarose gel is taken from seaweed. And also remember, the DNA are not moving above it. The DNA are moving within it, through it. Okay, that is why it is said their sieving effect is being shown. So there is sieving effect, and that is a very important question as well. Okay, so you might have to pick the wrong statement. They might give you that the DNA moves on the surface of the agarose gel. No, within the agarose gel, that is the correct thing. Small details are really important. Now, okay, so once you have separated, what do? You take a nitrocellulose sheet, you put it like this. Now everything comes over here on this particular sheet. Now you put ethidium bromide. Very important, very important. What do? Ethidium bromide you put. Now you think that it might be visible. No. Then you have to treat it under UV radiation. My dear children, when you put it under UV radiation, what will you observe? There will be bright orange colored bands. There will be bright orange colored bands over there, my dear children. Okay, under the UV light, only under the UV light. So if you are just treating it with ethidium bromide, that's not enough. You have to show it under what? UV radiation. Now the separated bands of DNA are cut out from the agarose gel. What do? After you got it, okay, you found that okay, they have separated and things like that have happened. Now slowly you will cut those bands from the agarose gel, and basically you are going to take it out. Separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This piece is known as elution. This step is known as elution. What do you call this step as? When you are separating this out and taking out, this is known as elution. The DNA purified in this way are used in constructing recombinant DNA technology. Now the question that have come from this particular field is that name the stain used to visualize DNA fragments in gel electrophoresis, ethidium bromide. Choose the correct sequence of gel electrophoresis. The cutting of DNA by restriction enzyme, staining by ethidium bromide, exposure of UV rays, separation of DNA fragments under the influence of electric field in agarose gel. So what is the thing? The cutting of DNA by restriction enzyme, then separation, then staining, then exposure to UV radiation. That is how you do. Okay, so if you do not know the steps, how will you be able to answer this? So that is the thing. Okay, now talking about the vector. So you spoke about how do you separate with the help of the electrophoresis. You also spoke about restriction enzyme. Now it's time for vector. The vector is the one that will be carrying the thing. Okay, carrying the gene of interest. So what all features it should be having? A proper origin of replication, my dear children. It should be having an origin of replication. So it is a place where replication starts, and it is also the sequence which is responsible for controlling the copy number of a linked DNA. For example, how many copies we want? That is also dependent on the origin of replication. So origin of replication are two functions. One is where the origin replication starts, and the second is where it controls the copy number also. So you have to choose a vector, or you have to choose a thing that has origin of replication that supports high copy numbers as well. A piece of alien DNA cannot multiply itself in the progeny cells of the organism due to a normal piece of alien DNA which is just there in a in the progeny cell is not able to multiply itself. Why? A piece of DNA which is just there in the progeny cell, it is not able to multiply, probably does not have origin of replication. That is why it is not able to increase its number. That is the thing. Okay. Now the second thing that a vector must have is selectable marker. What do you mean by selectable marker? So selectable markers which helps in identifying and eliminating non-transformants and selectively permitting the growth of transformants. What do you mean by transformation? I told you initially. For example, I am a Escherichia coli. Inside this Escherichia coli, I am putting another organism's plasmid. Now, I have become a transformed Escherichia coli. Okay, that means I have undergone the process of transformation. How do other organisms understand whether I am transformed or not? Okay, it would be only understood if I have a selectable marker within me. If I have a selectable marker within me, that's when it will be able, very easy thing to understand. So selectable markers come usually in the form of antibiotic resistance. So for example, I am an Escherichia coli, I have a plasmid that has resistance for ampicillin as well as tetracycline. Now what do? The gene of interest which is there, it is put in my tetracycline resistant area. So now I am no more resistant to tetracycline, my dear children. I am not any more resistant to tetracycline. That means I am transformed. I am the one that has undergone the process of transformation. So transformation is a procedure through which a piece of DNA is introduced in a host organism. Now, resistant to antibiotics such as ampicillin, chloramphenicol, tetracycline and kanamycin, etc. are considered useful selectable markers for E. coli. So just read through it, the antibiotic names are really important. Okay. Now, cloning sites. In order to link the alien DNA, the vector needs to have very few preferable single recognition sites for commonly used restriction enzymes. So this is PBR332, my dear children. Very E. coli has this vector. Okay. E. coli cloning vector PBR E. coli cloning vector PBR322 showing restriction sites. These are the restriction sites. Okay. So listen to me very carefully. So over here, this is a restriction site for tetracycline. This is the restriction sites for ampicillin. But okay, not this. Oh my God, what happened to me? I'm so sorry. Not the restriction site. This area means it has a genes which provides resistance to tetracycline. That means if I am putting antibiotic that is tetracycline, it will be resistant to it. It won't have any effect. Why? Because it has a tetracycline resistant gene. But in the same gene, I have two sites. One site is for BAMH1, a restriction enzyme. Another site for another enzyme, that is SAL1. If suppose the BAMH1 is active and it makes a cut over here, and now I am going to put my gene of interest over here. Do you think it will be still tetracycline resistant, my dear children? It would not be tetracycline resistant because I have inserted something else over here. You get it? Now it will show me, okay, since I have my gene interest inserted over here, this won't be resistant anymore, and hence I can select this particular gene. I can select this particular plasmid or this particular organism which has the plasmid. That is why it is known as selectable marker. So the ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. Over here I have ampicillin and tetracycline both. I will take one and I am going to put my gene of interest over one. Foreign DNA at the BAMH1 site of tetracycline resistant gene in the vector. Okay, the recombinant plasmid will lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out from the non-recombinant ones by plating the transformants on the tetracycline containing medium. What I am going to do is that, okay, I put it over here. but still I am not able to understand. So what do? I am going to put it in a medium that has tetracycline, and then I will understand, because it won't be able to survive the tetracycline resistance, right? Anyways, it is not resistant anymore, so it won't be able to survive tetracycline, it will die off. You get it? There will be changes in them. So that is how I would be able to identify. The transformants growing on the ampicillin containing medium are then transformed on a medium containing tetracycline. The recombinants will grow in ampicillin containing medium, but not that on tetracycline. So the recombinants which are there, it will be growing on ampicillin since it is having the gene, but it won't be growing in tetracycline medium because it does not have the gene. The recombinants, the one that has the gene of interest, my dear children. So that is there. Now the question is asked, what is insertional inactivation? So there also you saw that in the tetracycline gene, if I am inserting something of my choice, that activity is lost. It is not resistant to tetracycline anymore. Similarly, any gene, if it is losing its function, okay, or is it getting inactivated because I am putting my gene, this is known as insertional inactivation. So there is another example. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta-galactosidase. So there is an enzyme beta-galactosidase. Instead of this particular coding sequence of this enzyme, I am going to put my gene of interest. This results in inactivation of the gene for synthesis of the enzyme, which is referred to as insertional inactivation. Now this gene is not produced. What happens if this gene is not produced? What is the use of this anyways? The presence of a chromogenic substrate gives blue colored colonies if the plasmid in the bacteria does not have any insert. Okay, so usually if there is no insert, it will give blue color. Okay, why? Because there is a chromogenic substance being added. When it is added, the beta-galactosidase which is there, it acts on it and converts into blue color. Presence of insert results into insertional inactivation of the beta-galactosidase genes, and the colonies do not produce any color. These are identified as recombinant colonies. So now what happens? Since this enzyme is not there, I have put my gene of interest. Even if I add the substrate, it won't be giving me blue color, it will give me white color. That means this colony of organisms have my gene of interest. They are the recombinant one, they are the new ones. I have to select them and clone them and increase their number. You get it? Again, this is a selectable marker. Okay. So the enzyme responsible for breakdown of X-gal is the chromogenic substance, okay, is beta-galactosidase. The cells containing recombinant plasmid will produce the cells containing recombinant plasmid, the one that has recombinant. That means the beta-galactosidase is being removed, and instead of that you are putting your gene of interest. That will produce what? White colonies, my dear children. Because why? Beta-galactosidase is not being produced. Since beta-galactosidase is inactive, whatever you want to say, you can say. This type of questions comes from these paragraphs. Okay. Now over here again there are certain names of the vectors given. So please remember, whenever you are talking about plant, for plant you have a very well known vector, that is Agrobacterium tumefaciens. It is a bacteria, okay. It has a plasmid which is known as TI plasmid, tumor inducing plasmid, and this plasmid has a specific segment that is known as transfer DNA, which is responsible for causing tumor in plants. Now what do I do? Anyways, this is going to infect. So if I remove the transfer DNA and put my gene of interest, it is still going to infect, but it is going to give the gene of interest only. So isn't it acting as a vector? Isn't it acting as a way through which I can give my gene of interest to the organism that I am looking forward for? That is a plant probably. So Agrobacterium tumefaciens is a pathogen for plants, but it is also going to act like a vector only if it is disarmed. Disarmed as in I am removing the transfer DNA within the plasmid. The plasmid is known as TI plasmid, tumor inducing plasmid, because it has a transfer DNA. I cut the DNA, transfer DNA, and put my gene of interest and ask them to work. Okay, so in that way they will be normal. So the commonly used vector, okay, now the tumor inducing plasmid of okay, has now been disarmed and modified into cloning vector, which is no more pathogenic to the plants but is still able to use mechanism to deliver genes of interest into variety of plants. Now, retroviruses have been disarmed and now used to deliver desirable gene in animal cells. So, what is the vector for animal cells, my dear children? It is retrovirus. Please do not forget, retrovirus for animal cell. Name the plasmid present in Agrobacterium tumefaciens, TI plasmid or tumor inducing plasmid. Tumor inducing plasmid. Okay. Now the last thing, and then I think we are done. Okay, I think we are done with that, with the chapter itself. So listen, so you have made recombinant plasmid and everything, you have put the gene of interest and everything is there, but you have to put it in the host also, right? Host organism, you have put it in the vector, agreed. You have put it in the vector, and this vector is going to be the one that is going to carry out its work. but now you have to put it in the host where the actual product is going to be produced. How do you do it? Okay, you have to make it competent. You have to make it what? Competent. So first thing, understand that DNA is hydrophilic, that means it hates water. Sorry, hydrophilic as it is love water, but it cannot pass through the cell membrane, because cell membrane is what? Hydrophobic in nature. At least the head region is hydrophobic. That is why they cannot, obviously the DNA cannot move through. Now, to make the cells competent, you can do three things. Okay, first thing, this is done by treating them with a specific, them as in cells, with specific concentration of divalent cations such as calcium. So what do? You treat it with divalent cations, that is calcium, my dear children. It increases the efficiency with which DNA enters the bacterium through pores in its cell wall. So there are pores made in the cell wall because of the action of this divalent cations, that is calcium. Now after that, what do? You incubate the cells with recombinant. Recombinant DNA is there and the cell with the pores is there. Okay, now what are you going to do? You are going to put it on ice. You are going to put both of them on ice, followed by placing them briefly at 42 degrees Celsius, that is heat shock. Now you are going to give heat, and again you are going to put it back into the ice. In this point of time, what happens? The recombinant DNA which is there, it enters the cells through the pores and get inside, and the pores then get closed because of the ice. This enables the bacteria to take up the recombinant DNA, my dear children. Okay, so first then you treated them with specific concentration of divalent cations such as calcium, which increases the efficiency with which DNA enters the bacterium through pores in its cell wall. How do you make the pores? You make the pores through this, not through the calcium. You are going to make the pores through this. Why? How? Basically you are going to treat with ice, then with heat, then again with ice. This is known as heat shock treatment, my dear children. This is usually done for the bacteria to take up the DNA. Now in the case of animals, what do? You do micro injection. In micro injection, there will be tube-like thing. Okay, you are going to inject the DNA directly into the nucleus. So micro injection here, the recombinant DNA is directly injected into the nucleus of the animal cell. Now suitable for plants, the next thing is biolistics or gene gun. Micro injection for animals, this is very, very important. For each there is different. For bacteria, heat shock, then over here micro injection for animals. Now for plants, what do we have? Gene gun or biolistics. Where what do? You have a gene gun like thing where there are microparticles of gold and tungsten being used, and it is covered by a DNA choice of interest, whatever is there. And now you are going to put it like inside, you are going to shoot it with high speed, with high velocity inside the nucleus. So that, why are you doing it? Because you know there is a cell wall in the case of plant cell, which is why you have to use a gene gun there. That is the way you do. Okay. Now there is one more way, use of disarmed pathogen vectors, which when allowed to infect the cells, transfer the recombinant DNA into the host. You can use the disarmed vectors like retroviruses or probably any bacteria, and then you can with the help of that also you can transfer the gene of interest. Disarmed as in they are not going to cause you any trouble, they are going to just help with the transfer of DNA. Now plant cells are bombarded with high velocity microparticles of gold and tungsten coated with DNA in a method known as what? Gene gun, or what do you call as biolistics. So this is the thing that you need to know. And rest of the process we already studied it, my dear children. I know this is kind of confusing now, and I know I might also have made you confused at some point of time, probably while talking about the vector. So if you need a clear explanation, I can make a 5 to 10 minutes video on how does that vector thing work. Okay, I can surely do that for you, but you have to tell me in the comment section. So that's all, my dear children. Nothing else, nothing else, nothing else, nothing else. So yes. So if you think that this particular video was useful for you, do not forget to subscribe to the channel and also like the video. And also if you have any doubt, please do not get scared away and not ask me anything. Please ask me. Okay, these comments and all in class and all, doubts can be asked face to face. Here there is no option for that. That is why I am asking you, if you have doubts, do not keep it to yourself. Your identity and identity, you are just behind your phones, my dear children. Okay, you don't have to worry about who will judge you, and why should someone judge you for the first thing? Even if they know you, why should be they judging you for the doubts that you are asking? You should be freely asking your doubts. Doubts are the one that gives rise to what invention, that is what is said, right? So definitely do not forget to ask the doubts as well in the comment section. So that's all. Take care. Bye bye.